Great. Thanks very much for inviting me. It's a huge honor to be here. I'm an ophthalmologist based in London at Moorfields Eye Hospital and St. Thomas's Hospital. I'm Omar Mahroo, and I specialize in retinal disease, particularly inherited retinal disease, and electrophysiology. And my research is based at University College London. So do come and visit London sometime, and I can take you around Moorfields. I'm going to talk about advances in electroretinography. I always have far too many slides and have to rush through and no one knows what I'm saying. So I'll refer you to this review that I published quite recently, a few months ago, that has a lot of what I'll be talking about. So feel free to look at that. And I'm also happy to email my slides to anyone who wants it. So I called it Visual Electrophysiology and the Potential of the Potential. See, that's an electrophysiologist joke there. Good on you. So moving on to levels, so I just want to talk a little bit about why we need ERGs. These are levels at which you could probe the visual system. So these are the stages of vision, photons of visible light, all the way to the subjective perception of vision. And the way we normally, certainly clinically, and even in many research settings, but clinically, the way we investigate visual function is really psychophysically. You measure visual acuity or perimetry or other psychophysical tests, and there could be a problem anywhere earlier in the visual pathway that could impact on that. And obviously we have tricks, particular field defects tell us where the lesion might be, but really we don't directly assess function routinely of these other levels in the visual system. If we look at earlier levels, it's with imaging, and we have amazing, tremendous advances in imaging, retinal imaging that have occurred over the last few decades. And if we want to look past the retina, we have neuroimaging, but it's still largely structure. In terms of assessing the function of those levels in the visual pathway, then electrophysiology really allows us to do that directly. So VEPs from the cortex and the ERGs, different variations on ERGs, the electroretinogram, to directly assess signal processing and signals being generated in the retina. So I often say that we have this amazing, pretty imaging. It looks really pretty, but the retina is not just there to sit and look pretty on imaging, it's there to generate electrical signals. So we should have a way of quantitatively and objectively assessing the electrical signals from the retina, which is the function of the retina. This is how many of you know about ERGs, perhaps, I don't know, but this is how you record the ERG, very simple, just have an electrode in contact, preferably with the front of the eye or near the front of the eye, and another electrode behind the eye, and just measuring the potential difference, and you're looking at the change in voltage in response to light stimuli. That, believe it or not, is a picture of me over 20 years ago during my PhD, and when I had more hair. And a few years ago, I was at Arvo in Vancouver, and my PhD student came to me and said, "Your picture's on someone's post." I went to the guy, said, "Oh, that's me." And he kind of looked at it and looked at me and said, "Nah." So I said, "No, it is." He said, "I got it off the internet." I said, "Yeah, I uploaded it onto the internet." Anyway, so that's how you record the ERG. And this is, many of us are familiar with retinal structure as you can see on OCTs, you have outer retina and inner retina. These are the main players in terms of generating the visual signal, photoreceptors, bipolar cells, ganglion cells, and many other cells modifying transmission. Photoreceptors effectively turn off with respect to life. They become more light, not life. They become more negative. Voltage becomes more negative, so you could call them off cells. And bipolar cells, many of them depolarize in response to the stimulus from the photoreceptor, the signal from the photoreceptor. They're on bipolar cells, and many hyperpolarize, and they're off bipolar cells. And they're the main generators of the flash. Deliver a flash of light, you get a downward deflection and then an upward deflection. So the downward deflection is basically photoreceptors hyperpolarizing, brackets also off bipolar cells, and the upward deflection is largely the on bipolar cells. So as long as you can tell up from down, you can interpret ERGs, and you can, literally, we had lots of high-level talks this morning. This is just at the level of the retina. You can get at what the photoreceptors are doing and the bipolar cells without taking someone's eye out, just with electrodes around the eye. And the other stimulus we often use, that's flicker stimulus, a sensitive indicator of cone system function. and you deliver flashes in the dark, you get the rod system, and in the light, you get the cone system. It's basically showing the same thing, but with, you know, arrows and things. Photoreceptor, you have this circulating current in the dark, a flash of light generates the phototransduction cascade, shuts off that circulating current, the cell hyperpolarizes, that gives you most of the A wave, and then glutamate transmission is reduced at the synapse, and the depolarizing currents are set up in the bipolar cells, and that gives you much of the B wave. And so we know a lot about where much of the ERG comes from, so it's a nice tool to investigate retinal function. If you have a problem at the synapse, either side, you selectively reduce the B wave, and you get what's called an electronegative ERG. That's a very helpful clinical sign as well. The International Society for the Clinical Electrophysiology of Vision is there to standardize ERG testing protocols. So in the old days, everyone just doing different things, and you couldn't really compare between labs. But now we have an international society, and they set a minimum protocol, and that's been really helpful. This is another picture of me, not so long ago, but when I still had more hair. I was sent to Tanzania. We had a link between London and Tanzania, Dar es Salaam, Vision 2020 link to sort of help development of ophthalmology there. And I was, they'd been donated an ERG machine from a Japanese eye department. So I was a registrar, I just said, well, go off you go, and you know about ERGs, why don't you go and set it up? I got there, and you can see that the manual was in Japanese, right? It's come from Japanese eye department, but I don't speak Japanese. So I've flown all the way to Tanzania, but luckily I could read the numbers, and the numbers all corresponded to the ICEV protocol. So we got it working. I don't know how useful it was, but we got it working anyway. ICEV, as I say, sets the standards for, that's the latest standard for full field electroretinography. If you're interested to know about ICEV standards, then there was a good guide published in 2018. To visual electrodiagnostic procedures, and these are the standards that are in the literature, the latest updates, and again I can send the slides, you'll have to go through them, but for full field ERGs, multifocal ERGs, and then ICEV have these extended protocols for taking things beyond standard testing and telling you how you might assess other aspects of retinal function beyond the standard. And then almost all the way into my talk, we're now going into the main subject matter, which is where next. So what I want to now talk about is recent and anticipated future advances in electroretinography in terms of devices, stimulus protocols, novel analyses, the potential that's contained within those waveforms, and then a couple of examples if we have time. So in terms of devices, one major advance is portable devices are now developed, have now been developed. Actually, they've been around for decades, but now we have portable devices within the last decade that have cameras incorporated, so you can see the subject's eye, whether you can. It has a pupilometer, so as you know, standard ERGs need to be done with pupil dilation, which blurs the patient's vision for hours. This has a pupilometer and adjusts the stimulus intensity accordingly, so you get the right retinal illuminance. So I think that's a huge advance. Within a few minutes, whereas ERG testing normally takes an hour or more, certain clinical and scientific questions, I think, can be answered within minutes with an ERG, where you don't have to dilate the subject's pupil or wait for dilation, and just do a light adapted ERG and get some answers. So I think they have been a bit of a game changer. Even there are companies developing ways of using a smartphone to generate and record an ERG. That's not really widely available. And also multimodal devices. I think it'd be great to be able to stimulate localized parts of the retina and get the ERG. We have the multifocal ERG, but that's still relatively coarse. There are devices that have been developed that are OCT guided to just stimulate certain localized regions of the retina and record responses from them. And I think that would be transformational in our assessment of efficacy of gene therapy, where we've put a new gene in replaced a gene in a particular part of the retina, these blinding inherited retinal diseases, to see really is that working or not? Is that area of retina now working? There have been many papers published with the handheld ERG, portable ERGs, and this is something we published relatively recently, looking at quite a rare disease called birdshot chorioretinopathy. In that condition, it's quite hard to know whether you should go or down on a patient's treatment, and the ERG gives you some hard data by which to guide your treatment. Except in many centers, it takes months to get an ERG. Patients have to be sent to another hospital. Whereas if you have a handheld ERG in clinic, you can get one of the most important parameters within minutes. So again, I think that's something that's been transformational for these patients. And there are some other conditions as well, where I think a quick few-minute ERG can really tell you what's going on without having to wait months, and the patient comes and has their pupils dilated again and spends hours on another visit. Other advances are in terms of stimulus protocols, rather than just some flashes delivered in the dark and flashes and flickering light delivered in the on the light background. We have these extended protocols that I mentioned, and then going even further, because standard protocols generally, most of them, not all of them, most of them just use white stimuli on a white background. Yet there's so much more information. Christopher Tyler's doing spectral ERGs, as are others, trying to look at spectral aspects of the stimulus and selectively record from different cell classes. Separating rod and cone contributions to dark adapted responses has been a problem. There's no perfect way, but I'll speak in a couple of minutes about a way in which we do that and the things you can tell from that. And also tracking adaptation of the retina. So ERGs classically are done in the dark, patients dark adapted, so for 20 minutes, and it's a steady state, and then you put them in the light for 10 minutes, it's another steady state. Whereas many patients have problems going from light to dark or dark to light. That's their main thing. Once they're in a particular state for a while, they can see all right, but it's the adaptation that's affected or prominently affected. And we don't really have very good ways of measuring that. We have psychophysical ways, but they can be demanding on the patient. Whereas the ERG can be used as a sort of pan-retinal objective way of looking at adaptation, changes in retinal sensitivity on different backgrounds, with the patient not having to do much, just sit and look into a bowl and not necessarily press a button and work out whether they've seen something or not. And again, I'll talk a bit more about that in like a few more minutes. And then the other main area of advances are in terms of novel analyses. Rather than just looking at amplitudes and peak times, people have looked at waveform transformations, applying mechanistic models of phototransduction to the ERG, so that you can, from those models, extract parameters that relate to human phototransduction from the living eye. I spent a lot of my time applying mathematical models to ERGs. When I tell people I work in modeling, they sort of look at me a bit funny. Really? And I say, no, mathematical modeling of ERG. Oh, yeah, you look like the kind of person who'd do that, yeah. And AI methods. So AI is being applied to just everything and every aspect of medicine. So we've applied, and others have applied, machine learning methods to electrophysiology, where you may be able to get an automated, so ERG expertise is relatively scarce, but if you had an automated way of triaging patients or categorizing them, that could be very helpful, make ERGs much more accessible. But also further than that, rather than just matching the performance of experts, I think AI could be used to extract subtle features of the waveform that are not discernible to the human expert, no matter how many ERGs you've looked at. Just as from retinal photographs, an AI algorithm can tell if the patient's male or female. And I've looked at thousands of patients' fundi, and I can't tell from a fundus whether someone's male or female, but obviously there are easier ways of telling. But the fact that the AI algorithm is noticing something in the image that we as human experts, the greatest retinal experts, still can't kind of fathom what that is very interesting. And then combining ERG data with genetic data, looking at ERGs are often used in rare inherited retinal disease, but we record in sufficient numbers of people. We can even look at small genetic variants, potential modifiers in various diseases, common genetic variants, and look at their effect on the ERG, which I'll also talk about, and also get insights into myopia, which I may or may not talk about. And then the retina is effectively a bit of the brain, isn't it? It's the most anterior part of the brain. Much of the circuitry is similar to the circuitry of the brain. So the recording ERGs with novel protocols, I think, can give us insight not just into the visual impairment in certain neurological conditions, but in terms of even mechanistic insights into those diseases. This is again another recent paper where we applied machine learning to ABCA4 retinopathy. Stargardt disease. ABCA4 is the commonest genetic cause of inherited retinal disease, which is the commonest cause of blindness in the working age in many countries. And we found that a machine learning algorithm could correctly pretty much categorize patients into different electrophysiological groups. And even more than that could, again, I say we, it was actually Sophie Clinton, the first author there, developed a nice ERG-based classification of genetic variants. There's over 1,000 genetic variants in ABCA4 in Stargardt disease, and we get bewildered by that. We don't really know how to counsel the patient in terms of what the risk of loss of peripheral vision is, but now we have an ERG-based score by which to score those variants. Just briefly on as I was saying, these are dark adapted, light adapted ERGs. This is what we normally measure. Peak times and amplitudes and a little kind of hand waving about the shape of the waveform, but there's so much more in that, in those simple waveforms. Even the standard waveforms, I think, I won't go through all of these, but you can tell, kinetics of phototransduction, synchronization between on and off bipolar cell pathways, retinal ganglion cells. There's a lot more hidden in those waveforms that perhaps we need an AI algorithm to extract for us. And then finally, just in the last few minutes, I wanted to give some examples. So I talked about separating rod and cone contributions. You have flashes in the dark. Their traces are responses, average responses from a human subject, different flashes of different intensity delivered in the dark. And they, as I said, that's the photoreceptors hyperpolarizing, giving those A waves, but they come from rods and cones, and it's quite hard to split them up. One way in which we do that is we apply a blue background of sufficient strength to pretty much saturate the rods, but hardly at all desensitize the cones, the L&M cones, which is the majority of the cones. And then that can give you responses, dark adapted responses from the cone system, and you simply mathematically subtract those traces from the other traces, and you get the rod component of the response. And that's a way we've used in many different studies to try and work out what the rods and the cones are doing separately in the dark. Again, I won't go through this in detail, but these are the stages of phototransduction, and Lam and Pugh 30 years ago, amazing it was that long ago, developed a mechanistic model to describe the kinetics of phototransduction and the change in circulating current in response to light. And interestingly, that same expression can be applied to the ERG, the A wave bit, and it fits really well. A single equation with just different intensities of parameters for intensity of light fits pretty well. And then later, Robson and Frischman have refined that model so that it actually fits the A wave trough and the initial recovery as well. And we're getting many insights into inherited retinal disease from different inherited retinal diseases from fitting those models. Even in myopia, we published this recently last year. Myopia is increasing worldwide. No one quite knows why. No one knows what's driving myopia. What we do know is it's the retina, and many of the genetic associations with myopia seem to be expressed in the retina. And we found an interesting association. One of the strongest associations with myopia near a gene encoding gap junctions in the retina and cone-driven off bipolar cell responses. And we use some of the techniques I talked about to come to those conclusions, but I haven't got time to go to that into more detail. And then finally, tracking retinal dark adaptation. Again, this is a review published last year of physiology about using the ERG to track retinal dark adaptation. This shows the visual cycle. You don't need to see it till just that there's lots of different steps and it's very complicated. And we use the ERG, I'm running out of time, so I'll go very quickly. We use the ERG to develop a new model for photopigment regeneration. And when we went back to densitometry data, that's the normal way of looking at pigment regeneration in the retina. And these are some previously published studies and the way in which those authors fit their data. When we applied our ERG-derived model, it fit the traces far better. So I always love that when a predicted model goes right through the data. I call that my happy place. We've shown that also for rod system recovery as well. So because I'm running out of time, or I've nearly run out of time, I won't go through the rod system adaptation, but again, the ERG can be used to look at kinetics of rod dark adaptation, and there are some different conditions, vitamin A deficiency, Sorsby fundus dystrophy, even age-related macular degeneration, where I think these the ERG could really be applicable to categorize those diseases and give us new insights. And the final thing that I'll just skip over was we also used the ERG to explain this phenomenon of transient smartphone blindness. These were patients complaining of unilateral night blindness who were had all sorts of investigations done, and all it was they were lying on one side at night with one eye being covered by the pillow, as we all do, and viewing their smartphone. So one eye was dark adapted, and the other eye was light. That's all it was, but they had all sorts of investigations. And even now I'm getting emails from patients all over the world that I think this might be what's wrong with me, but my doctor's changed all my medication. And all it was is simple physiological adaptation. And we showed it with the ERG that this is, I did it in my own eyes and viewed a smartphone and showed that it's just retinal, rod dark adaptation demonstrable at the level of the bipolar cells. The media went crazy and said, oh, smartphones cause blindness. No, we're trying to say that. I was telling you about how the ERGs can tell us about disease. This is an example where the ERGs can show us that something isn't really a disease, but we could prove it with the ERG. So I've run out of time, but those are my concluding remarks. There are continuing advances being made, as well as in imaging in electrophysiology. We have tools by which to understand normal retinal physiology and pathophysiology. And I haven't talked much about neurological diseases, but I think we can get insights into those as well. And we have widening accessibility with portable devices and AI-assisted interpretation. These are some recent relevant papers from our lab, but I'll end there and happy to take any questions or emails. Thanks.
Well, thank you for that whirlwind tour, Omar. Is anyone sufficiently compos mentis to ask any questions? Yeah.
It seems to me that assumes that the rods are linear, and another way to do this would be to flash the rods with something that doesn't excite the cones but saturates the rods. And if you do that, you should get the same thing if the rods are linear. Has anybody done that? And are the rods linear?
It's a brilliant question, and yeah, there are different. There's loads of things I skipped out in that talk, as you imagine. Yeah, there are different ways of trying to do that, and none of them are perfect. And one is the way I showed, which is the easiest when you have patients, rather than giving a bright flash and then immediately another flash. But the technique you describe is also used, where you deliver a bright flash. It'll end up stimulating the cones as well, but they recover within a second. So then you deliver the next flash that you're interested in, and you know that the response to that one's only coming from the cone system, and then you subtract that out. And other methods are using photopically or scotopically matched red flashes or blue flashes, but it's a bit complicated because you have to iteratively subtract them. But the way I showed isn't perfect, but it's probably the easiest when you're doing it with patients. Yeah, they do. So the one way where we think we probably are doing the right thing is they all give broadly the same results. Yeah. Welcome.
Could you describe the handheld ERG system, whether that's a commercial device or something you developed?
Yeah, so I didn't develop it, and I don't get any cut from the company, so I'm very comfortable talking about it. It's called the RetiVal, and it's produced by LKC Technologies. It's not perfect. It's maybe the conventional systems have less noise, but very convenient, easy to use. They have their specially developed skin electrodes, so you can do it with skin electrodes or with conventional ERG thread electrodes or others. And it is commercially available and quite responsive. The company, if you have a particular protocol you want to put in, they at least with me, they've sort of changed it so that I think with Christopher as well, so that you can do that. But it's a very nice way of getting ERGs within minutes, clinical or remote setting.
Actually, oh.
Yeah, so beyond like the regular colors of like red, blue, and green, are you aware of like studies that have done and looked at near infrared light for like photobiomodulation?
Right, that's very interesting. So there are some studies that have looked at near infrared light in terms of enhancing mitochondrial function. Is that what you were thinking about?
Yes,
Yeah,
yes.
and in terms of directly assessing then effect on the ERG, I don't think that's been shown. Where I'm based, the Institute of Ophthalmology, is another PI, Glenn Jeffrey, who's very interested in the effects of infrared light. And they did retival flicker ERGs and found that some people, the peak time became earlier and some it didn't. And to my mind, it didn't really show anything significant. I don't think he thought it was significant either. And maybe you need larger numbers or something a bit more controlled. But yeah, it's an interesting question.
Thank you.
Thanks.
Thanks very much, Omar.